Hello anatomy students! Today we're going to talk about the events that occur at a synapse to transmit an action potential from one neuron to another. The two processes that make this possible are synaptic transmission and summation. Here are the learning objectives. We'll first differentiate between two types of synapses and explain how electrical signals travel from one neuron to another. And then we'll talk about graded potentials, which are also known as postsynaptic potentials, or PSPs. We will learn how they're generated, how they're spread through the cell membrane, and how they affect transmission of an action potential down a neuron. So here on this slide, I have two pictures of synapses. The one that's on the left is called the electrical synapse. And as you can see in the picture, we have a physical connection between our presynaptic cell and our postsynaptic cell. And that physical connection, this is like a tunnel that's built by proteins, and these tunnels are called gap junctions. And these gap junctions allow the cytoplasm from the presynaptic cell to mix with the postsynaptic cell. So it's almost like they're one giant cell. And this is going to allow for the flow of ions to be continuous from one cell to the next. And they don't have to undergo all of the steps that happen in the chemical synapse. So the chemical synapse is what I have pictured on the right. In a chemical synapse, we have two cells. We have our presynaptic cell, which is here, and we have our postsynaptic cell, which is down on the bottom. But unlike in an electrical synapse, these two cells don't physically connect to each other. Instead, they're separated by a very small gap, which is called the synaptic cleft. You'll also notice that our presynaptic neuron, the one that's on the top of the picture, has a lot of synaptic vesicles or bubbles that contain neurotransmitter. And that our postsynaptic cell, or the postsynaptic membrane, is kind of dotted with these receptor molecules that are specific for the neurotransmitter that's housed in our presynaptic cell. So electrical synapses are fewer in number in our bodies than chemical synapses. Our electrical synapses are found in the olfactory bulb, in the retina, and also in our central nervous system. But the chemical synapses are going to be more common. Now, even though textbooks usually depict synapses occurring between the synaptic knob on a presynaptic neuron and the dendrite of a postsynaptic neuron, I just want you to know that there are other synaptic arrangements that we have in our nervous system. So the first one is called axodendritic, and again, that's where the axon of the presynaptic cell makes a synapse with the dendrite of a postsynaptic cell, and that's what you have in our light purple box here on our slide. But we also have axosomatic synapses, where the axon from the presynaptic cell is going to synapse with the cell body or the cell soma of the postsynaptic cell. And then finally, in the tan box, we have our axoaxonic synapse, where the axon of the presynaptic cell synapses with the axon of the postsynaptic cell. The axoaxonic arrangements allow one neuron, are going to allow the presynaptic neuron, to either augment or inhibit the conduction of an action potential that has already started traveling down the axon of the postsynaptic cell. Okay, so what is actually happening at the synapse? Well, first of all, when an action potential reaches the synaptic knob of our presynaptic cell, that's going to stimulate calcium ion channels to open. And calcium ions are going to flow into the cytoplasm of the synaptic knob. This calcium influx is going to cause the vesicles, which is here at number 2, to merge with the interior of the cell membrane and exocytose the neurotransmitter into the synaptic cleft. And so that's what you see here. You've got all the pink little neurotransmitters leaving the cell, and now they're entering into the synaptic cleft. At number 3 in our picture, the neurotransmitter is going to diffuse across the synaptic cleft, and it's going to bind to the receptors, and it's going to either directly or indirectly initiate a local potential, a local postsynaptic potential. And then here at number 4, the postsynaptic potential, it may move to the axon, and it could help to generate another action potential to travel through the axon of this postsynaptic cell. So those are the things that are happening during synaptic transmission. A postsynaptic potential, also called a graded or a loco potential, is a change in the membrane potential in the area associated with the receptor. So where the receptor is embedded in the cell membrane, that little area around it, that's the part of the cell membrane that is affected by a change in the membrane potential when a neurotransmitter binds there. And then what happens is it radiates outward from the area of the receptor. The further the postsynaptic potential gets from the point of origin, the weaker it becomes. And the reason for this is because the cytoplasm of the cell acts as a resistor to the spreading of the potential. In the picture that we have here, if you look at the bottom bar graph, you can see that stimulus 1, that's shown in green, has the least amount of strength, and that stimulus 2 has the middle amount, and stimulus 3 has the greatest amount. And if you look at the graph above, the black line is showing us the resting membrane potential. And you can see how the membrane potential changes. As the strength of the stimulus increases, the amount of change in the membrane potential also increases. Postsynaptic potentials can be excitatory or inhibitory. Excitatory postsynaptic potentials, which are also known as EPSPs, they're going to cause a local depolarization. So in order to cause that local depolarization, in order to cause our membrane potential to become more positive, it's going to cause sodium and some potassium channels to open. And that's going to cause sodium to enter into the cell and a little bit of potassium also. And that's going to cause a temporary depolarization in the area of that of the receptor for that neurotransmitter, and then it's going to radiate outward from that point. Excitatory neurotransmitters are the ones that are going to cause excitatory postsynaptic potentials. And excitatory neurotransmitters include dopamine and norepinephrine. An inhibitory postsynaptic potential, or an IPSP, occurs when an inhibitory neurotransmitter like GABA, the long word for that is gamma aminobutyric acid, which acts in the brain. Another example of an inhibitory neurotransmitter is glycine, which acts in the spinal cord. So when these inhibitory neurotransmitters bind to a receptor, they're not going to cause depolarization. They're going to cause hyperpolarization. They're inhibitory. So they're going to cause potassium channels to open, and potassium will flow out of the cell. And it can cause chloride channels to open, allowing chloride to enter from the extracellular fluid into the cell. both of these things are going to cause the membrane potential to become more negative, and it's going to cause a temporary hyperpolarization. And so if you've got positive potassium flowing out of the cell and you have negative chloride flowing into the cell, well, that's just going to drive our membrane potential to be more negative. So I wanted to show you on this graph what's happening to the membrane potential when an inhibitory neurotransmitter binds to a receptor on the neuron cell surface. And so as you should remember from our study of action potential, the resting membrane potential is here at negative 70 millivolts. And when an inhibitory neurotransmitter binds in the area of that receptor, you're going to get potassium channels to open, and potassium flows out of the cell. And that's going to drop the membrane potential, making it more negative even than the resting membrane potential. But then, as I said on the previous slide, some of these inhibitory neurotransmitters will also cause chloride channels to open, and that allows negatively charged chloride to enter the cell as well. And that's going to make it even be more negative here. So again, because we are dropping our membrane potential lower than our resting membrane potential, what we have here is hyperpolarization that is happening here. So this is hyperpolarization. And so that's what's happening here because we have our membrane potential dropping below our resting membrane potential. When we have a neuron that is in this state, nope, I lost my curve there. When we have a neuron that is in this state, we can also call this neuron inhibited. Because if it's down here where we have our hyperpolarization, if we get a stimulus, say right here, usually a single stimulus is only going to raise our membrane potential by 0.5 millivolts. And so 0.5 millivolts is not going to be enough to raise this membrane potential all the way back up to a negative 55. Like that's where it would have to go. Because remember, negative 55, like negative 60, that is threshold. That's what has to be met in order for an action potential to occur. So we say that our neuron is inhibited because it's going to take quite a large stimulus in order to knock it out of hyperpolarization and to get it up to this negative 55 area in order for an action potential to occur. So this is not going to happen very often, and so we say that the neuron here is inhibited. So as I said on the previous slide, like a single stimulus is only going to raise the membrane potential by 0.5 millivolts. And so it's just important to understand that a single stimulus cannot cause an action potential in a neuron because that 0.5 millivolt change does not allow the membrane potential to reach threshold. You need about a 10 millivolt change or so in order to raise the membrane potential from resting to about negative 60, which is 60 or 55, which is about the value of threshold. And so I think that it begs the question, how are action potentials generated? Well, they are generated through a process that's called summation. It's an important process because it's going to allow neurons to kind of decide which impulses we receive should not be ignored and which ones should be ignored. Because we are receiving stimuli from various sources every moment of every single day. And we can't possibly process it all. We would just be overwhelmed. And so only the ones that cause action potentials are recognized by our brains. The gatekeeper, the one that's keeping track of all of the part of our neuron that's keeping track of all of these impulses that are coming through, is the axon hillock. So if all of these impulses create a membrane potential around threshold at the axon hillock, then yeah, we're going to get an action potential being propagated down that axon. If not, if at the axon hillock they do not come together and combine in order to reach threshold, then an action potential will not occur. So there are two kinds of summation for you to know: spatial summation and temporal summation. And as I discuss these two, I'm going to use excitatory postsynaptic potentials, or EPSPs, as my examples. So the first type of summation for you to know is called spatial summation. So remember, a neuron receives stimuli from many other neurons. So even though in pictures in the textbook, they show like one synaptic knob and they show one dendrite, it's really like one neuron is receiving input from multiple different neurons. And that's exactly what's happening in our diagram here. You can see that we have stimulus 1 over to the left, and then stimulus 2 is down here at the bottom. So we have two different synaptic knobs that are transmitting signals to our neuron. Now, these stimuli in this picture are arriving at the same time, but in different locations on the cell membrane. This is called spatial summation. So each stimulus has its own space. And so what you can see in this picture is that the stimulus that's being measured in this area has this magnitude, and then the other stimulus that's being measured over here has a magnitude that's shown on the little voltmeter screen. And when they reach the axon hillock, you can see that the peak, the strength of that stimulus, they've added together, they've summated, and now they have a taller peak. They've added together. And that's what we're seeing kind of here in this different picture. We have two different axons that are giving a signal to our cell membrane. They are adding together. Threshold has been reached, and now we'll have action potential propagation. So again, spatial summation means that we get multiple EPSPs at the same time on the postsynaptic neuron, but they are occurring in different places. So they travel and they combine at the axon hillock, and if they reach threshold, then we'll get an action potential. If not, then we won't. Now, could this have been an EPSP and an IPSP? Sure, and I'll get to that in a couple of slides. Temporal summation is the second type of summation for you to know. In temporal summation, you have a rapid succession of stimuli that are arriving at a single synapse. So now we only have one green axon knob that is synapsing with our neuron here. And what we're getting is multiple stimuli from that one axon, from that one green axon, and they are all traveling towards that axon hillock. And so here in our top picture, we have two different stimuli, and they're coming together, and then when they're added together at the axon hillock, you can see that the peak in the voltmeter is taller. So again, when they combine at the axon hillock, if they have caused the membrane potential to reach threshold, then yes, we are going to get propagation of an action potential. But if not, then we won't get an action potential. And so, like I said before, what if EPSPs and IPSPs reach the hillock together? Will an action potential even occur? Well, it depends on the strength of the EPSPs and the IPSPs. And it's kind of like adding positive and negative integers together. So in my example where I'm adding a -3 to a +7, our answer is a +4. So when you're working that operation, our positive number, the 7, is larger than the negative number, the 3, and so our answer is positive, positive 4, because our positive integer was bigger than our negative integer. So it's the same thing. If the EPSPs are larger or stronger than the IPSPs and threshold is reached, then yeah, we're going to get an action potential propagated down that axon hillock. If the IPSPs are larger or stronger than the EPSPs, then hyperpolarization at the axon hillock will occur and an action potential will not be generated. So what happens then to our neurotransmitters after they bind to the postsynaptic cell and they cause EPSPs and IPSPs to happen? Well, there are a number of different things. They can be transported back to the synaptic knobs from whence they came, and they can be reused. They can be metabolized into inactive compounds by enzymes that are present in the synapse, and then those pieces can be reused by the presynaptic neuron. Or they can even diffuse away from the synapse and they can be used by other cells in the body for other purposes. All right, let's see how much you remember. So number 1, which synapse physically connects cells? Your answer is the electrical synapse. That's the one that has protein tunnels that are going to physically connect our pre- and postsynaptic cells. Number 2, which ion triggers neurotransmitter vesicles to the plasma membrane for release? If you said calcium, you would be correct. Number 3, which type of postsynaptic potential causes hyperpolarization of the postsynaptic cell? If you said IPSP or inhibitory postsynaptic potential, then you would be correct. And finally, what kind of summation occurs when multiple stimuli arrive at the neuron in close succession and combine to produce a large depolarization? That would be temporal. Yes, I am correct. I lost my mind for a minute there. That would be temporal summation. So because if it were spatial, then the multiple stimuli would have to arrive at the same time. Okay, anatomy students, thanks for listening. I hope you learned something, and I'll talk to you in class.